tissue fixation. Fixation, we are looking at histology samples, and the major aim is to maintain the tissue in a lifelike state. So in front of us we have two histological samples. One of our sample is breast in regard to tissue fixation. Number two, the kidney. Fixation is the process of preserving, we are preserving biological tissues in a lifelike state. Okay? And the major purpose why we are fixing this tissue is by stopping what we call autolysis and putrefaction. So autolysis, that is enzyme digestion, whereas putrefaction is bacterial action on tissues. So upon blood interruption, it's very important that we immediately fix biological tissues. And commonly we use 10% neutral buffered formalin as a common histological fixative. So in other words, the fixative is a substance which preserves the shape, structure, relationship, and chemical constituents of tissue or cell after death. So upon blood interruption, in other words, these cells or tissues are cut off blood, or there is no blood supply to these tissues. So since there is no blood supply to tissues, it means there is no oxygen anymore, and also no nutrients. Tissues are no longer receiving nutrients. So usually there are changes that occur after death, and that's what we call post-mortem changes. Fixation is aimed at stabilizing the structure and the chemical composition of cells or tissues, making them suitable for microscopic examination and long-term storage. So as we have said, we have our two samples. So when you look at this sample of ours, this is a breast tissue, but it is in what? It is in fixative, 10% neutral buffered formalin. The essence is to arrest post-mortem changes. So generally, post-mortem changes, these are physical, they can be chemical or microscopic alterations that occur in tissues and organs after death. Why? Number one, due to cessation of blood circulation, because upon blood interruption, these tissues are no longer receiving blood. Number two, cessation of respiration, metabolism, and defense mechanism. Example, one of the changes generally is that upon blood interruption or when somebody dies, there is cooling of the body, okay? So after death, metabolism stops, okay? And the body temperature gradually equilibrates with the environment. So cooling rate depends on environment. It can depend on clothing. Number three, it will depend on body fat. And number four, depending on cause of death. You've heard that when somebody dies, the body becomes cold. Number two, we have post-mortem rigidity. So this occurs 2 to 6 hours after death and may last between 24 to 48 hours. And why? This is due to depletion of ATP, okay? Therefore, the actin and myosin filaments in muscles remain irreversibly bound, and that results into stiffness. You'll hear that when somebody dies, the different parts of the body become stiff. Number three, hypostasis, okay? So hypostasis, we are looking at gravitational pulling of blood independent body parts after saturation stops. And that makes the body appear purplish red, okay? Majorly of the skin and the organs. And this begins within 30 minutes to 2 hours after death. Then the other change, autolysis, which you call self-digestion. So autolysis is caused by release of lysosomal enzymes from the dying cells. Okay? So, and it is more rapid in enzyme-rich tissues like the pancreas. We can talk about the stomach, brain, and the liver. On microscopy, there is loss of nuclear detail, cytoplasmic homogenization. Number 5 is putrefaction, which you call decomposition by bacteria, mainly intestinal flora. So these bacteria, upon death, they produce gases like hydrogen sulfide, methane, and ammonium, which causes bloating, foul smell, greenish or black discoloration. So histologically, we're going to see massive tissue destruction and bacterial colonies. Change that occurs is hemolysis of red blood cells. So upon death, red blood cells break down, they release hemoglobin, and hemoglobin diffuses into surrounding tissues, sometimes mimicking hemorrhage. So we are going to see a pink staining of vessels and tissue around the blood vessels. Cooling of the body helps in estimating time since death. That one can be used in forensic studies. And also post-mortem rigidity, the forensic indicator of time since death. Hypostasis helps differentiate between antemortem bruising and postmortem settling. Whereas autolysis is very important since it may obscure true pathology if fixation is delayed. And usually putrefaction, when there is advanced decomposition, is a forensic indicator of longer interval since death.